So good evening. Today is your week 9 session with me. So please confirm that am I audible and are my slides visible? Thank you. Yeah, so in week 9 session, we will be, just give me a moment. Yeah, so in this session, we will be discussing about the introduction to proteogenomics, and we will also read about sequence-centric proteogenomics, gene variant analysis, and the role of proteomics in clinical studies. So in today's session, we will be mostly practicing MCQs, and with those MCQs, we will be looking into these topics as well, and we will look into the current literature. And if you have any doubts regarding the assignment, so you can ask them also in between. Yeah, so today's session will entirely be the MCQ kind of thing. So um, it will be better if you will also respond to these questions because it has to be interactive session. So the first question is, what are the effects of genome sequence variation on the proteome level? And the options are, the sequence of protein changes, protein modification site does not change. Option C is protein sequence does not change, but protein level increases or decreases. And option D is no effect on the protein level. So you have to tell that what is the effect of variation in the sequence of a genome and what impact it has on the proteome level. So you can type on the chat box or you can also reply. Okay, A and C. Yeah, right. Yeah, because we know that if our genome sequence will change, then there are chances that protein sequence can also change. Whereas in other scenarios, what can happen is that if the sequence is changing from the known coding part, that means which is not giving rise to protein sequence, but it is in the known coding region. So it can have a regulatory effect, which can control that in which level the protein will get expressed. So yeah, option A and C are correct. Whereas protein modification site does not change. So it is not always the condition because in certain scenarios, what can happen is that if amino acid get changed, so it can also interfere with the protein modification sites because there are certain amino acids which are attributed to certain type of post modifications, means post translational modifications. So if those amino acids will get changed, so in that scenarios the modification won't take place, and in the same way modification site will get changed. So in that way option B was not correct. Similarly, no effect on protein level, so we cannot say that always there will be no effect because there are chances where protein levels can also change. So that's why option D was also not right. Yeah, so there was a review published in the Journal of Molecular Biology in which they have talked about the impact of variations in the human genome on the gene regulation. So in this, they have clearly explained that how the changes in the sequence of the genome can impact the protein sequences or protein levels. So in this, they have mentioned. So I have just taken the few takeaway messages from this paper. So in this, they have mentioned that with the advances in sequencing techniques, now it is possible to attain the sequence of DNA and RNA, and we can also perform large-scale studies of human genomic and transcriptomic variations. So with this, we can also identify the type of variations because genomic variations are of multiple types. They can be single nucleotide changes or they can be insertions, deletions, and structural variants. And these variants can also affect the protein sequence as well as the level. And in some scenarios, even if they don't affect the protein sequence, then also they can manage the or they can regulate the expression of a protein. So the genomic projects which are kindly attributed towards these capturing these variations are 1000 Genome Project and the ENCODE. So their main purpose is to identify the human genome sequence. And then after comparing multiple human genome sequences, they will be able to capture the genomic variations and they will also be able to identify the functional elements, that which are the elements of the genome which are involved in the functionality, either protein functionality or regulatory roles. So all those things are identified by these projects, and then they identify the impact of these variations on the proteome. So the variants can alter the protein sequences or they can modify the protein levels, which means that these variations will either be on the promoters or on some regions which are kind of either cis regulating or trans regulating the protein expression, or there are chances in which there will be no effect on the protein sequence or protein levels. So there are some cases where this is also possible, and protein levels may change without sequence variation depending on the regulatory variants. Then genomic and functional data. So integrating genomic variations with transcription and functional data is key to understand how these variations affect the gene expression and the proteome. So if you want to read in detail, then you can go through this review and it will easily explain. So this was a very short review in which each and every point was clearly explained, that how the change in sequence is influencing the proteome expression. And then there was another article which was published in the Nature Perspectives. So in this also they have mentioned, so they have designed a complete consortium, which is the impact of genomic variations on functions. So other than the experimental information which is already available, what they are doing is they are also doing the predictions that how a certain variation can lead to some functional consequence. So their main purpose is to decipher the impact of genomic variation on the function. So in this, they have mentioned that these genomic variations, they nearly influence all the aspects of human biology, which are also linked to the health and disease. So that's why this study of identifying the DNA variants which are linked with the function is very important, so that we can address the issues which are influencing the human health. So in this, they have designed a consortium, which is the impact of genomic variation on function, which is IGVF consortium, which aims to map that how these genomic variants they affect the genome function and the phenotype. So this will produce the open resources which will link the genetic variation to biological and disease outcomes. So as we have seen in the previous lectures that how NCI has designed the uh, cancer data sets and how those data sets are open, all the repository information is collected at the same place. So how data commons have been developed. So similarly, their work is also the same. So they are also designing a open source platform where everyone can access this kind of information. So in this, they are generally using the data of single cell mapping, genomic perturbations, and predictive modeling. So the new thing which they have incorporated into their method is the predictive modeling. So just other than the experimental evidences, they are also trying to predict, and then they are just checking that how much their predictions are right. So with that, more and more predictions can be made. So across hundred of cell types to achieve this. So in this figure, they have shown that how the genomic variations can be captured. So firstly, across individuals and populations, the information of genomic variants is captured. So these variations can arise from single nucleotide variations, or these could be indels, which means insertions or deletions. So single nucleotide variation will be the change in the single nucleotide position. And there could be multiple SNVs present in the human genome, but it means that when we are only looking at a single nucleotide change, whereas insertion and deletions they could be on the larger nucleotides. So then we call them as indels. And then there could be some structural variations which are also possible. For example, certain indels can lead to the structural variations. So that is, and translocations or transpositions, they can also lead to structural variations. So then linking the genome variation to genome function, because we already have the human genome information. So in that way, we can link them to the genome functions, and these functions could be the regulatory elements. So how those variations are influencing the function, with that we will be able to identify the regulatory elements and how they influence the gene expression. And if gene expression is varied, then how it will influence the protein expression and activity. And ultimately, as we know that multiple proteins which are present in the human body, they interact with one another to perform some function. For example, as we know in our different pathways or different digestive system or any other respiratory pathways, they all involve multiple proteins which are working in a connection to one another. So in that way, multiple proteins interact with one another and they constitutes a network which is known as molecular network. So we also call them as PPIs, which means protein-protein interaction networks, but here they are calling it as molecular networks because metabolites and other means constituents are also present. So that's why it's a journal term to mention that there are molecular networks which can get changed. For example, if you change the protein sequence, so if its functionality is changed, for example, if its active site is changed, or if there is any change in the protein sequence which is leading to the loss of its functionality, so in that way it won't be able to perform in particular pathway. So if it is coming in between of a pathway, then other links which are present downstream of that pathway, they will also fail to perform their functions because they will not get the output of that particular protein. So in that case, molecular networks will also get perturbed or disturbed, we can say, and ultimately they will influence the cellular phenotypes. So all these informations can be gathered from the genome function comparisons, and these are mostly done with the already established data sets which are already available. And then they are linked to the phenotypes, that how a particular variation is influencing the phenotype. For example, if these SNVs are linked to one particular type of cancer. For example, I'm taking normal person's genome and a cancer patient genome, and then I'm comparing and I'm identifying the variations or mutations. So in that way, not a single comparison can lead me to these conclusions. But if I'm doing a big level study, for example, for a metagenome or for multiple large scale studies. So in that case, I can conclude that these variations are linked with this particular type of cancer. So in that way, we can link these variations to the phenotypes. That what are the variations which are involved in the healthy individual and what variations lead to the disease risk. And then we can also judge that if we distinguish between different stages of cancers and then take their genome, then we can also distinguish between the severity of the variations of the disease. That these variations are leading to the severe disease conditions. And then we can also be able to identify the treatment response, because different people can react in different ways to different drugs. So in that way, that is the main concept, that why the proteogenomics came into play, so that we can design the personalized medicines. So which means that each person, although the DNA is similar to some extent, but still everyone reacts different to different drugs. So all those things can be captured from the phenotype associations to the genomic variations. Then what are the applications of this? So fundamental and disease biology. So firstly, we can clear the fundamentals. We can understand all these things, that how they are linked. But the second thing, which is the main application of this association, is genetic diagnosis. So by looking at certain variations or SNVs, indels, or structural variations, we can simply diagnose that some variations can be the disease specific variations. So even if we find them, we'll be able to identify that this is the particular disease which the person is affected with, and along with this, even we can also identify the stage, that at which stage that person is in the particular disease. For example, in cancer. Then risk predictions. We can predict that whether that disease is severe or it is known severe and it can be treated. We can also manage the clinical things. So we can manage that which drugs must be given at this particular level. Similarly, target identification and therapeutic development. So this is also one and the same thing, that if certain mutation is linked to a disease, so that can act as a target, and we can design the drugs so that which can interfere with that particular mutation and remove it or cure it or do something to treat that disease. So this was about the entire aim of their this perspective article. Then later on, they have mentioned the flow chart of their framework. So they have mentioned that they have designed the map perturb predict framework, in which what they have done is they have taken the data from single cell mapping, which will be taken for each and every cell, which is RNA expression data and chromatin accessibility data. So after taking this data, they will be able to prioritize the cell types and assays. So what does prioritize cell types means? That in our body, there are certain cells which are more prone to certain kind of diseases. So in that way, they will be studying those particular cell types, and then their RNA expression will be mainly studied. So in this, they have used means there are already some repositories which capture these informations, because they are not themselves doing all these experiments. So there is 10x, Multiome, ShareSec, and Parse. So these are already available data repositories which contain these kind of informations. So what they are doing, they are capturing this single cell mapping information, and they have prioritized the cell types and assays, and then they have identified the perturbation targets. So what perturbation target means, that they have identified the genes which are more prone to variations or modifications, and they are linked to certain kind of disease. So they have identified those target genes or target proteins, so that they can make some changes in those genes and proteins. They can perturb them, which means they can themselves do some kind of modification and then check the functional outcome. So for this, they have for genomic perturbations, generally single cell RNA-seq studies are used, and then SN attack-seq, chip-seq, VAMP-seq, RNA flow-fish, cell sorting, and cell painting. So these are already available experimental techniques which are generally used, and then the effects are captured. So what they are doing is they are perturbing certain kind of genes, and then they are performing these experimental studies and checking that what is the effect of that perturbation. So this is just to design a data set. So some informations are already collected and some informations are further on checked. And what kind of perturbations they have done is base editing. They have changes some kind of nucleotide bases, or they have prime editing. They have done prime editing, or MPRA, star sec, CRISPR. I means interference, or A means activation. So they can either inhibit some gene, or they can activate some gene, or overexpression. So they can also overexpress some gene. Overexpression means that gene will overexpress and it will form multiple RNAs, and ultimately it will synthesize more amount of protein. So in that way, these are the types of perturbations which they have done on the selective cells and selective genes. And ultimately, they have measured their effect by using these experiments. So now this is the dataset generation. This is just done to train the model. So now if all these perturbations have been done, then they prioritize the perturbations. So for example, if some perturbations are leading to major changes, for example, if in cell painting, what they do is they check for the morphology of cells. So if some variation is having the major change in the uh, morphology of a cells, so those variations will be captured first. They will be prioritized. So now what they do is they prioritize those perturbations and they train and evaluate models. So now in these models, what will be done is that for every gene, all the variations which have been done, they are kept, and then their associated outcomes, which are captured by these methods. So all those informations are captured, and then these models are trained. So whenever new variation is assigned, now whenever the new input is being given to these models, they will make some prediction that what is the possible impact this variation can have on the functionality of a protein or physiology of the protein. So all those things can be now predicted. So this is known as predictive modeling. So this is based on the training set. Training set is generally developed to train this model so that if new variations is inputted into this model, then it can predict the future outcome. So now what kind of biological questions which can be answered by this, or what questions they have mainly asked is the coding variations. So in coding variations, they have asked that which missense variations of unknown significance. So VUS stands for variations of unknown or uncertain significance are likely to be pathogenic. So there are some variations which are missense. So whenever we study the single nucleotide polymorphism or single nucleotide variations, so there can be mainly there are mainly two categories. One are synonymous variations and one are non-synonymous. So synonymous variation mean whenever an amino acid is whenever there is change in single nucleotide position, the amino acid which it is giving rise to, that does not change, that remains the same. So as we know that in the genetic code, whenever we see the triplet codons, they give rise to amino acid. So it is possible that multiple triplet codons can give rise to same amino acid. So in that way, even if a single nucleotide gets changed, still it can give rise to a same amino acid. So in such scenarios, that is known as the sense mutations, or we can say synonymous mutations. Whereas missense mutation means whenever the change in a nucleotide position happens, so it leads to change in the amino acid sequence. So which comes under the non-synonymous mutation, which means the mutations which do not give rise to similar type of amino acid. So in non-synonymous, there are two further categories. One is missense mutation and another is nonsense mutation, which are also known as the stop mutations, we can say. So in that, what happens is single nucleotide change leads to the formation of stop codon, and ultimately it will stop the protein extension, and it will lead to premature termination of the protein. So those mutations are known as nonsense mutations. So in this case, they have mentioned missense mutation, which means single nucleotide change giving rise to change in the amino acid in the sequence of protein. So firstly, what can we identify by this? So we can identify that how many such missense variations of uncertain significance. For example, till now their significance is not associated. We don't know that what this mutation is giving rise to. For example, in the previous lecture also we have seen that there are certain mutations which are simply passenger mutation, which do not have any influence on the cancer, which is not helping the cancer to grow or metastasize. So such mutations, to understand their role, that whether they could be pathogenic or not, that can be identified by using this predictive modeling and by using their method, which is IGVF. And then non-coding variations. So which variants affect the expression of which genes? So that also we can means find out. If there is some variation in the non-coding region, then how it is influencing the expression of that gene, its regulation. So that can also be carried out. Then molecular networks. So with this, the molecular network information can also be captured, that how the interaction between two protein get influenced. So for example, in this case, as you see, ChIP-seq. So ChIP-seq is chromatin immunoprecipitation. So this is also done for the protein-protein interaction analysis. So that can also be used to identify that how particular mutation is leading to change in the interaction between two particular proteins and how they are influencing the molecular network. So then next thing is the disease. So which variant, cell types, genes, and pathways influence the disease risk? That can also be answered by using their technique, which is of predictive modeling. And this is the bigger flowchart of their IGVF catalog. So in this, they have clearly mentioned. So these purple lines represent the IGVF experiments. So whatever information is taken from experiments, that is mentioned in the purple color, and then in red color, they have mentioned the predictions which they have done. And in IGVF predictions exploratory, that is mentioned in the orange color, and in blue color, external integration. For example, if they have taken data from some external databases. So in this entirely, if to explain it in a short way, I can say that they have not just done their own analysis, but they have also taken the help from the external databases such as DBSNP, GenomeAd, HPRC. So means they have used some information from other databases, GTech and Code, and they have combined all those variant informations to identify the role of those variations in genomic functions, such as how they influence the cell type and cell states, what are the regulatory elements where the mutations are happening, and how it is influencing the functionality of that protein, means how it is influencing the expression of that protein, we can say. So attack seek is like this is also to check that in which sites which are more prone to the binding of transposase, and they establish, they tells that which sites are most active. So if the sites will be more active, that means they will be more available for the transcription factors and they will be more active. Their expression will be more. So in that way, attack seek is generally done for that, to find out that which genes are highly expressed and which genes are not expressing to that extent. Then single cell RNA-seq is done for the sequence level analysis, and means from a single cell. And for protein, UniProt is used. So this tells about the sequence of protein, and UniProt is already established database which contains the information of a protein sequences along with other informations such as their function, their structural information is also present. So this is also kind of a common repository which contain the information from all other servers as well. And then for molecular networks, already available gene ontology and INACT. So these are already available databases which contain this information. And they have done PPIs, scRNA-seq. So these are the experimental studies which are done to identify the molecular network. And then for the cellular phenotypes also, information is for cellular phenotype, mostly experimental things are done. They have not mentioned any such available database from where they have collected this information. So ultimately, they are telling that how they have designed this IGVF catalog, how all the variations are linked to different diseases, how they have identified. So this is just a flow chart that how they have gathered it. And ultimately, they have linked those genomic variations with the phenotype and disease. And ultimately, here they have mentioned that how they are doing. So they are benchmarking, they are integrating, and they have done the predictive modeling. So for predictive modeling, for benchmarking, they have taken the experimental studies which are already established, and then they have integrated both the already available experimental studies as well as their perturbations and its predictions. So means benchmarking is simply done to check that whether your model is doing better or not. And then we can take the new input set and we can make the predictions if our model is performing better. And now they have mentioned that with time, their model is performing better. So in the y-axis, they have mentioned the accuracy and completeness of their particular catalog. So yeah, it's with time it is improving, that they have mentioned. So this is all about the IGBF catalog. So is it clear? Okay, thank you. So now let's move to the next question. So in this, they have mentioned that Shantanu wants to build a protein database for the cervical cancer. Which of the following things will be required for the protein database generation? So now he wants to design a protein database. So option A is sequence of the gene. Option B is position of the gene. Option C is sequence of the protein. Option D is location of the protein in the cell. And option E is 6' translation data. So you have to tell that what of these, which of these points are most important to design a protein database. The option D. So D with D you mean this option 4, right? Okay. Yeah, so you're right. For developing a protein database, we simply need a sequence of gene, sequence of protein, and its 6' translation. And the position of gene is not required because we want to develop a protein database and position does not matter. And similarly, location of protein is also not important for designing the protein database. Okay, so now next question is, if Shantanu wants to develop a robust machine learning basis predictive model for his cervical cancer project, then what major challenges he might face during the predictive model building? So as we have seen in the previous literature also, they have done the predictive modeling only. So option A is small sample size, option B is random matching, option C is specific selection of data sets, option D is only A and B, and option E is A, B, and C. So in this, you have to tell that if somebody wants to develop a robust machine learning model, which is doing some predictive analysis or is predicting something, then what are the major challenges which someone face while designing those models? Okay, option E. Yeah, you're right. So first challenge is that small sample size. So if sample size is small, then for training, it's very difficult to train with the small sample size because you don't know that means in the global what is the influence because small sample size can be biased, it is not that much random, so with small sample size we are not confident enough with our model predictions, so that is one of the challenge. Second is random matching. So if sample size is very small and then if there is random matching, so that can also be a problem, so we have to verify that whether those matches are significant or they are just a random match. Then specific selection of data sets. So this is also important. If your data set selection is not proper or it is poor, then also it will not give you the proper results. So all these points are important. And these are kind of the challenges which someone face while designing some predictive models. Okay, so let's look at the fourth question. By building a proteogenomic database, what information Shantanu can get about the cervical cancer? So option A is it will provide information about the protein movement inside the cell. Option B, it will provide correlation between the genotype and phenotype of the cervical cancer. Option C, it will provide information about protein-protein interactions. And option D, it will provide information about the novel peptide sequences originated due to the mutation in the gene. So with the help of proteogenomic database, what kind of information Shantanu can get? Okay, correlation between the genotype and phenotype of cervical cancer. So yeah, this option was also true, but other than this, proteogenomics can also help us to get the information of protein-protein interactions as well as the information about the novel peptide sequences which originate due to the mutations, because we know that in proteogenomics, what we can do is we can also design the customized databases, because otherwise if proteomic study is done, so in proteomics we are limited by the already available reference databases, because after mass spec we require some reference database where we do the mapping of our spectra and then we map to the particular already available sequence. So in certain scenarios we might get the novel peptide sequences and we will not be able to map. So proteogenomics is designing those customized peptide sequences and helping us to identify the novel peptides. So proteogenomics provide us that advantage that we can get the information of novel peptide sequences and which might get originated due to the mutations in the genes. So these all are the three important informations which Shantanu can get. But if this was not the MSQ, which means multiple selective question, if it is a MCQ, then the right option will be option B because it is most favorable option in these out of these four. Yeah, so now let's look at the next question. So which of the following order is correct for a regular SILAC experiment? So these are different steps. First is desalting, mix N14 light and N15 heavy in 1:1 ratio. Option 3, cell lysis and digestion. Option 4, confluent cell line in N14 light media. Option 5th is replace N14 light media with N15 heavy media. And option 6 is mass spectrometry run. So these are some of the steps which are followed during the regular SILAC experiment. So you have to organize them into a proper order. So I think SILAC was covered in the session. Yeah. Okay, option C. Yeah. So this is the right option. Now we will quickly go through one of the literature in which they have means mentioned that how the SELAC is used. So in this, it is entire protocol that how different steps are there. So we will see so that you can remember these steps if you will go through this paper. So this is simply a practical recipe in which they have mentioned that how the stable isotope labeling by amino acids in the cell culture can help you with multiplexing different samples and then studying multiple samples at the same time. So major things which they have mentioned is that SELEC labels the proteomes metabolically. When we say metabolically, which means that there will be some amino acids which will be labeled with some isotopes which will be stable. For example, what kind of elements, or we can say what kind of atoms which will be isotope labeled. So they can be carbon, nitrogen, and hydrogen which are present in the amino acids. They can be labeled. So what are these isotopes? So for carbon, normally it is C12, but isotope of C12 is C13. Similarly, for nitrogen, normal nitrogen is having N14, whereas in this condition it is having N15. Similarly, for hydrogen, it is H1, but in this case, it can have the isotope can have H2, which is the deuterium. So these are the different type of isotopes which are available and which are mostly labeled in the amino acids. So these heavy amino acids in growth media. So generally in the growth media, these heavy amino acids are supplemented so that our cells can get these heavy isotopes, and these isotopes get integrated into the proteome. Then mass spectrometry distinctions. So now if these heavy and light cells will be available, so they can be distinguished easily because the heavy cells will have different abundance and the light cells will have different abundance. So their signals will vary between one another. So in that way, we will be able to distinguish between both these light and heavy cells. And then they have also compared the SELAC with the chemical derivatization, that instead of using SELAC, if chemical derivatization is done, so what are the limitation of this chemical derivatization? So they have mentioned that SELAC offer precise quantification without the complexity of chemical derivatization, and it also avoid the extra modification steps and potential protein alterations. Chemical methods require multiple steps and may introduce errors because in chemicals you have to take different samples and you have to separately study those samples. So what can happen is during studying there are chances that you can incorporate some errors. So you can't rely on those results. Whereas in SELECT, on the single sample only, you can multiplex those samples and run them and in the same time. So that's why there are less chances of errors in the SELECT as compared to chemical derivatization. So firstly, in this they have mentioned that how quantitative information is encoded into the whole proteome with the SELECT. So firstly, subculture in heavy media. So here you can see these are the cells in the light, that is C612 lysine medium. This is the normal carbon. And then if its heavier isotope is present, so they have mentioned that if the media is supplemented with the amino acids which are labeled with this heavier isotope, which means carbon 13, then with some time, whenever the cells will grow, then in some time they will accommodate this heavier isotope in the cells. So whole proteome encoded with C613 lysine. So they will allow the cells to grow in that medium till the point all the means all the samples which are present inside it, all the proteins, they get changed with this heavy isotope. And after that, this heavy proteins will contain all those lysines because in the medium they have taken the isotope labeled lysine. So that's why in after means growing the cells, they will obtain all the proteins which will be labeled with these heavy lysine amino acids which will be present in that protein. Now they have performed lysine C digest in which the C terminal from C terminal positions lysine will be degraded. So there will be multiple peptides which will be generated. Now these will be the heavy peptides. So if at the same place another cell which is in the normal condition, if that is taken, so in that case the peptides that will be generated will be the light peptides, and then the analysis can be done and both the light and heavy peptides can be compared, and they have mentioned that in this case the difference between both these peptides will be of 6 daltons. So that is the mass to charge ratio, and it introduces a 6 dalton mass shift. So by incorporating this heavier isotope, the mass gets changed. Whereas if we talk about the isobaric labeling, so which we have seen last time, iTrack, TMD, those are the isobaric labeling methods. So in that case, the mass remains the same, but in SELECT, it is the isotope based labeling. So in this, mass gets changed. And with this change in mass, they can detect the differences between both the samples, light and the heavy samples. And in this, they have compared among chemical modifications and metabolic labeling. So they have mentioned that in metabolic labeling, so these lines in between, they are showing these samples are taken together. Whereas in chemical modification, the cells or tissues, they are separately chemically labeled, and then separately they are purified and fractionated. Separately, their protein is obtained, and ultimately their peptides are obtained separately, and then their peptides are compared, and then their MS sample is taken. So in this scenario, all these steps such as purification and the protein collection, they are done separately for each of these samples. So in this way, there are chances that it might incorporate some errors. Whereas in metabolic labeling, all these samples are taken at the same place from the beginning. So there are less chances of the errors. So this is only what this slide mentions, that in metabolic labeling, there are less chances and these are more reliable. Whereas in chemical modification, these results are not that reliable and these are also quite complex as compared to metabolic labeling. So that's why mostly metabolic labeling is favored. And in this, they have also mentioned that there are two phases of the select protocol. One is the adaptation phase, another is the experimental phase. So an adaptation phase means whenever our cells are adapting to the heavier isotope. So firstly, we start with some cell lines, some culture in DMEM, and then media with the light amino acid, which is the normal media, and then in the another cell. So in the another sample, there will be media with the heavy amino acid, which will be having the heavier isotope, which can be either carbon isotope, means carbon, which will be labeled with the heavier isotope, or it could be hydrogen, or it could be nitrogen. So anything could be labeled depending on your need, and there will be different differences which will come. So in case of carbon, we have seen there is a change in the 6 dalton, means 6 dalton mass is gained in the heavier isotopes. So it will vary among deuterium and nitrogen as well. So firstly, they have begun with the SILAC adaptation phase, in which they have supplemented the media with heavier amino acid isotope, and then they have subcultured these cells as needed, and they have cultured the desired number of dishes, allowing 5 doublings. So why they have taken 5 doublings? Because they have made sure that now means no one of these protein sequences present in the cell contains the isotopes which are lighter, means which are having the light amino acids. So in this case, they are making sure that the peak which they obtain, it is entirely coming from the protein sequences with the heavy labeled amino acid. So whenever this is obtained, the next step started, which is the experimental phase. Now in this stage, they have taken the control, which is state A, and which has the light amino acid, and then the perturbed or state B, which will have the heavier isotope, and they are mixed in 1:1 ratio. And then optional protein or peptide fractionation. So fractionation is done and other steps, desalting, all those things are done. And then ultimately mass spectrometry is done, in which their mass to charge ratios are calculated. And in this way, they both will be separated by some of the means mass range. So intensity of MS signal between light and heavy peptides give relative protein abundance between the cell states A and B. So you will be able to distinguish between both the A and B states, although even though you have mixed them and then done their MS analysis at the same time. So you will be able to distinguish. That is the main advantage of select. And in this, they have mentioned a protocol that after the peptide identification, if you does the ID match the raw data. So in this, they have mentioned that this is the raw data which is obtained after mass spectrometry analysis. Now there are two peaks. One is at 596.78, another is at 599.79. So you have to identify that which are the two peaks of light and heavy. So after obtaining those peaks, firstly what we have to do is we have to match between the this observed masses to charge ratio with the already expected masses to charge ratio. So we do the comparison so that we can provide the amino acid sequence. We can map the amino acid sequence or the peptide sequence to this particular spectra. So in that case, we match the ID with the raw data. If it matches, then well and good. But if it does not matches, then we verify the scan numbers and peptide IDs and mass to charge. So in that case, we have to search for another peaks in the data set. So if it does not matches the raw data, and if it matches, then we need to check whether it is a singlet peak. That means whether it's a singly charged or it is doubly charged. All those things are done now at this level. Then if it is a singlet peak, then it's fine. But if it is not a singlet peak, then you have to check that whether signal to noise ratio is good or it's not good. So signal to noise ratio specifies that whether your signal is better or your noise is high. So it is signal divided by noise. So if signal to noise ratio is high, which means you have the higher signal and lesser noise is there. So in that cases, we will favor it. Otherwise, if signal to noise ratio is poor, then we will again check and we will try to find another peaks. But if it is fine, then let's move to this step, singlet peak. So yeah, if it is a singlet peak, then we will check for the peptide ID and we will compare the peptide ratios against other peptides. And then ratios can be accurately computed for singlet peaks. And in that way, we can compare between the light sample and the heavier sample. Now in this case, if the signal to noise ratio is good, then we will check for the overlapping signals on either peptides. And if there are no overlapping signals, then we will compute the peptide XICs and calculate their ratios. And if there are overlapping signals, then will the narrower mass to charge window is computing XICs minimize overlap? Or are these other peptides for the protein with the usable ratios? So ultimately, we need to check that whether those spectra which are generated, whether these are similar to the expected spectra or how much it varies. So all those things are done just to make sure that there is not any noise which is getting into our results. It is the accurate result. It is the signal. It is the true signal. So these are just the steps which they have mentioned, and it is explained in this particular box. So you can read this box if you are interested. And ultimately, similar thing they have explained again, that how the analysis are done between the lighter and the heavier amino acid containing samples. So in this, they have mentioned two peaks and how much they are separated from one another. So in this, they have also mentioned that lysine is isotopically labeled with the carbon, as well as nitrogen is also labeled with the heavy isotope, which is N15. And then there is difference between both these peaks. So the difference is 8.01 daltons. And then they have mentioned the quantifying with XICs. So intensity is captured with respect to the retention time. And similarly here also they have checked that whether the signal to noise ratio is poor or whether it is better. So if it is poor, then we will move and we will check for the another peaks which are at the appropriate distance from one another and their signal is having the better signal to noise ratios. And ultimately the peptide sequences collected, peptide sequences assigned to that spectra. So this is just the detailed analysis. We don't have to go through this detailed analysis, but this is just for understanding. If you're interested, you can read it. Now let's move to the next question. Okay, so the question is, which of the following approach uses isobaric tagging? So now we have, till now we have studied isotopic tagging. Now you have to tell which approach uses the isobaric tagging. Option A is iTrack, option B TMT, option C SELAC, and option D both A and B. So we have already discussed this while talking about that paper. So what do you think? Which are the approaches which use isobaric tagging? Okay. Okay, let's see. So the answer is D, both iTrack and TMT. So TMT also uses the isobaric tagging, whereas SELAC uses the isotopic tagging. So iTrack, which is the isobaric tags for relative and absolute quantification, and TMT, which is known as tandem mass tags. So they both techniques they uses the isobaric tagging, and in these approaches, proteins from different samples are labeled with chemically identical tags that have the same mass, or we can say which are the isobars, marking them indistinguishable during their initial mass spectrometry run. Whereas if the second or the final mass spectrometry run will be done, then they will means uh, those tags they will generate different reporter ions. So you will be able to distinguish them, distinguish among both those samples which are labeled with the isobaric tags. So this is about iTrack and TMT. Yeah, now I have taken this um, you can say the categorization from this particular paper which was published in Frontiers. So in this they are very means properly explained that how the quantitative proteomics means what kind of methods are present under quantitative proteomics. So in mass spectrometry, as we all know, that we can also go for the quantification of proteins by looking at the abundance of peptide sequences. So in this relative quantification can be done. For example, if I want to compare among two sequences or two samples, so in that case, we want to go for the relative quantification. We want to go for the comparison. So in such scenarios, we either go for the label-based method or we can also go for the label-free methods. So under label-based methods, there are two techniques. One is metabolic labeling, another one is chemical labeling. So in metabolic labeling, SELEC and p-SELEC. So SELEC is stable isotope labeling by amino acids in cell culture, which we have already discussed. And then p-SELEC is the pulse SELEC, which is just a variation of SELEC, and it is used to track newly synthesized proteins over time. Whereas in chemical labeling, the methods are ICAT, which means isotope coded affinity tags. So it tags the cysteine residues for protein quantification. Whereas iTrack and TMT, they both uses the isobaric labeling. So they tag the peptides with identical masses that are distinguished during fragmentation, because they both will generate in the final run, they will generate different reporter ions. So in that way, they will be able to distinguish between two different samples. And then comes label-free quantification, in which spectral counting and precursor ion intensity. So they comes into play. In spectral counting, you count the number of mass spectra for a given protein to estimate the abundance. So in this, simply the spectras are counted. And the second thing is precursor ion intensity. So in this, you check for the intensity of a precursor ion, and in that way, by capturing that intensity, you predict that what will be the quantification. So if the higher the intensity, it is, that means it is it is in abundance, it is present in abundance, that's why its intensity is high. So in that way, you try to quantify. But overall, we mostly means favor the metabolic labeling or chemical labeling method, so better quantification instead of label free quantifications. Okay, now let's move to next question. So what is the primary goal of sequence-centric proteogenomics? Option A, to analyze the RNA sequences. Option B, to integrate genomic and proteomic data. Option C, to identify mutations only. And option D, to study the protein interactions. Okay, to integrate genomic and proteomic data. Okay, let's see. So yeah, you're right. So we use the sequence-centric proteogenomics to integrate genomic and proteomic data. So this is the primary goal. And it also enhances the accuracy of protein identification as well as genome annotations. And this approach, so other things also I want to discuss, that this approach refine the gene models, and they also identifies the novel protein coding regions, because we know that with the help of proteogenomics we can identify, because uh, refining gene models means, as we have discussed in previous sessions also, that in certain scenarios what can happen is that in the reference databases there might not be some uh, transcript variant information which is available, and now as with the mass spec we have identified some novel peptide which might belong to some of the region which was not already listed. So in that case, we might identify new exon or either new intron, which might be coding region or non-coding region. So in that case, we can means modify or we can refine the gene models. We can add new units to the gene models which are already available. So we can refine with the help of proteogenomics. We can also identify novel protein coding regions, and we can also detect the protein level variants such as single amino acid variants, splice junctions. So all these can be facilitated by sequence-centric proteomics, sequence-centric proteogenomics, and they also provide the personalized insights based on the genome sequences. And but it ultimately relies on the high quality protein sequence databases to improve the peptide identification and understanding of how the genomic variation translate into the functional proteins. So this is all about this question. Yeah, so I've also taken a mini review which was published in MCP, which is about the methods, tools, and current perspective in proteogenomics. So this was by the Kelly Ruggles group. So in the lectures also, she have already mentioned this again and again. So I have taken one of the section from this particular mini review, which was dedicated to sequence-centric proteogenomics, in which they have mentioned that how this proteomics helps to aid in the genome annotations, how it's linked to genome annotations, which can be done. And ultimately, with the help of proteogenomics, you can design the personalized sequence databases, because you can identify the protein level variants such as single amino acid variants, insertions, deletions, alternative splicing, novel gene fusions. So all those things can help you to design the personalized protein sequences. So these are just kind of the applications, you can say application of sequence-centric proteogenomics. You can also sequence the antibodies, viral infection and transposones can also be means their impact can also impact of viral infections and how it is influencing the activation of transposable elements that can also be studied with the help of proteogenomics. Then comes metaproteogenomics. So it focuses on the protein expression in complex microbial communities. So that's why it is referred to as metaproteogenomics because it is also involving the microbiome, which is already present in the either present in the human itself or in from the other sources as well. So all those studies also comes under proteogenomics. And then importance of protein sequence databases. So because if you have the sequence centering information, then you will be able to capture novel peptides or you can refine gene models. So all those things comes under the importance of the protein sequence databases. And then there was some visual representations in which they have mentioned that how these things are linked to one another. That from DNA you get whole genome sequencing information or whole exome sequencing, and from RNA you get the RNA-seq information in which RNA sequence is obtained. So you can combine both these informations to perform de novo read assembly or alignment to reference. This is ultimately done because after sequencing because now we know that there is advancement means we know Oxford Nanopore technique, Peg Biosciences, so they already means are available which sequence the larger because we can means with which we can sequence the entire genome in the one go. But still the primitive methods which were available, which was shotgun sequencing or all those methods, they were for the short stretch of DNA sequencing. So in that case we have to assemble that data. So means assembly is very much important so that you can assemble those reads and design a bigger or continuous stretch of genome. So then ultimately genome assembly is done in which multiple contigs which are either overlapping, they are means joined together to generate a scaffold and ultimately genome sequence means entire genome sequence is assembled. All those reads are assembled to design an entire genome sequence and then variant calling in which means because you are having the larger data set so you will compare among both of them. One will be the reference genome assembly which is taken as a reference which will be entire genome assembly and after that from these reads you will compare that where are the variations which are present which could be SNVs or all these variations. Ultimately customized protein sequence databases can be designed from these informations from these sequence informations. And then spectra and scoring and validation. So for scoring validation, you take the data from the proteins, proteomics experiments such as LC-MS, which is mass spectrometry analysis, in which the data is taken and then ultimately customized sequence databases and this spectra scoring and validation, these all can be combined. Ultimately, it will help to do genome annotations, mutation analysis, and metaproteomics, in which the peptides could be mapped to species as well. Because if the sample is taken from different sources, then you can also link those peptides to different species and you can compare their evolutionary history. So all those things can be done with the help of proteogenomics. So proteogenomics means they are integrating both the genomic aspect as well as proteomic aspect. So these are just the applications of this proteogenomic concept. I'm not means going to entirely discuss this because these are already covered a lot in the lectures. And then similarly, they have mentioned that how these expressions are linked to other means other data sets as well, such as EQTL expression, quantitative trait loci, or microRNA or copy number alterations. So all these things can be studied. So this slide ultimately shows that how the proteogenomics has advanced our analysis and what kind of information which we can get from this proteogenomics. So this just emphasizes that studying just genomics or just proteomics is not enough, and by integrating them, more insights can be generated. Now let's move to the another question. So in gene variant analysis, what do SNP stands for? So this is quite easy. Option A, standard nucleotide polymorphisms. Option B, single nucleotide polymorphisms. Option C, structural nucleotide polymorphisms. And option D is simple nucleotide polymorphisms. Yeah, so the answer is single nucleotide polymorphism. And we already know that these could be the changes at the single nucleotide position. Whenever a single nucleotide position changes, that is known as single nucleotide polymorphism, and it can occur both in the coding as well as non-coding region of the genome. and there could be substitutions, deletions, or insertions. So in this scenario, what we have seen that C is replaced with A, and in this also A is present. So we can either call it as C is replaced with A or substituted with A, or we can also say that in two organisms A is present. So might be A is substituted with C in this individual. Yeah, so these means SNPs could either be insertions, they could either be deletions, or they could either be simply substitutions. So in these scenarios, it is simply a substitution, but there are cases where it could be a insertion or a deletion as well. That will also be referred to as um, single nucleotide changes. And in polymorphism, it means whenever in multiple people, when we compare them, so that is known as polymorphism. That means in different individuals' genome, there is change in the sequence. So change in kind of morphology, you can say. So not exactly morphology, but change in the sequence. That's why it's referred to as poly, means multiple, and morphism means change. So if single nucleotide is changing among multiple individual genome, that is referred to as polymorphism. Okay, so now next question. What is a common application of proteomics in clinical studies? So option A, drug discovery, option B, genetic sequencing, option C, RNA synthesis, and option D, protein folding. So you have to tell the application in perspective of clinical studies. Okay. Yeah, so right answer is drug discovery. Yeah, right. Now next question is, which method is frequently used for protein identification in proteomics? PCR, mass spectrometry, gel electrophoresis, or Western blotting? So by now we already know, we have seen a lot of times this term coming again and again for protein identification. Okay, yeah. The right answer is option B, mass spectrometry. So in that we generally go for identification of proteins. PCR just for the amplification, gel electrophoresis is for the separation, and Western blotting is similarly for means getting different bands. So it is also kind of for separation. Okay, so next question is, which of the following can be a result of protein modifications? Okay, you are asking for the previous question. Okay, so yeah, I mean Western blotting, after that, if we go for the that particular step, then we can identify the proteins, but not for all the proteins we have already available. Uh, you can say the complementary molecules which can bind the protein of interest. So in that scenarios, we are not much able to identify the proteins. But it can be used if other antibodies, or in some cases some tags are also available, or some labeling, you can say. Yeah, means those are available for identification. But frequently used method is mass spectrometry, because since, as we have discussed also previously, that for all those proteins means antibody information is not available, so in that case we lag behind. So yeah, so which of the following can be a result of protein modifications analyzed in proteomics? Gene mutations, post-translational modifications, DNA replication errors, or RNA splicing? Okay, sorry. Yeah, so this is the right answer. Post-translational modifications, they are the result of protein modifications. Because gene mutations have no link with the protein modification, because they comes before the protein. And then DNA replication errors, they also comes before the protein synthesis. And then RNA splicing, so this is also the means this comes before the protein synthesis, before the translation. This is the RNA splicing comes under the post-transcriptional modification. Then comes translation, ultimately post-translational modifications. Okay, next question is, how can proteomics contribute to personalized medicine? Option A, by standardizing treatments. Option B, by tailoring therapies based on individual protein profiles. Option C, by eliminating the need for genetic testing. And option D is by identifying universal biomarkers. Okay. So the right answer is option B, by tailoring therapies based on individual protein profiles. So, means with the proteomics, you try to identify the unique or the individual protein profiles and then then design the personalized medicine. Whereas you cannot standardize treatments because we have seen that every individual respond differently to different treatments. So in that case, we cannot standardize treatment for all the people. And by eliminating the need for genetic testing, so no, because we need genetic testing to be done. So it does not eliminate that by identifying the universal biomarkers. So it's very difficult to find the universal biomarkers. It's difficult. But if we are able to find the universal biomarkers, it will be best thing. But it's difficult to identify the universal biomarkers because it might vary. But yeah, it can help us to identify the biomarkers which might be specific to certain kind of diseases, and in that way we can design the medicine. But in personalized medicine, it's difficult to identify universal biomarkers. That's why this option is not correct. If universal term is not included, then yeah, it will be a right option. Now question number 13. What type of study often employs gene variant analysis to assess the disease risk? Option A, clinical trial. Option B, cohort study. Option C, animal study. And option D is in vitro study. So in which of these studies we analyze the genetic variants to find out that how that variant is linked to a disease risk? Okay, option A you have said. Follow me, you want to make any guess? Okay, so you both are saying the clinical trials. So the right answer is option B, which is cohort study. And why not clinical trial? Because clinical trial is a term which is generally used when we are going for the drug testing. So whenever different individuals are given drugs, so clinical trial comes after that, in which there are different stages of this clinical trial in which it is checked that whether this drug is effective or whether it has some side effects. So whenever a new vaccine or any drug come up, so it has to go through this clinical trial stages so that we must be sure that if we are taking this drug, then it will not have some long-term consequence on us. So clinical trial comes under that thing in which the drugs are tested. Cohort study comes under the means when we have taken a small group of people, when we take it and we study that how a genetic variant is linked to a disease risk, or different studies means related to disease, if they are done on a group of people in which comparisons are done, that is referred to as the cohort study. And when we say animal study, so this is not exactly telling us, although the models in study can be performed on model organisms, but animal study is not sufficient to mention this. And in vitro study, so no. For gene variant analysis, we do not do any in vitro study because we want to study it on the people. Because in vitro we cannot replicate the same conditions. So that's why the option was also not correct. So if it is written as clinical study, then we might think of it as the right option, but it is written as clinical trial. So this term is entirely different from a clinical study. This is related to drug testing. So the right option is cohort study, and cohort means group of people, a small set of people in which some study is performed. That is referred to as cohort study. Okay, so next question is, in clinical proteomics, biomarkers are often used for what purpose? Option A, diagnosing diseases. Option B, synthesizing proteins. Option C, modifying DNA. And option D, sequencing RNA. Okay, so the right answer is option A, but I'm really confused why Pauline said option D. Okay, so the right answer is means biomarker studies or biomarker identification is done for the major purpose is to diagnose the disease, to identify that what disease is available. And another thing which can be done with the help of biomarkers is that if you know the biomarker for a particular kind of disease, then you can design the therapeutics which might affect that biomarker, which can means influence the activity of that biomarker so that particular disease can be cured. So first main purpose is diagnosing the disease. And then the second purpose is to design therapeutics related to that biomarker. Synthesizing proteins is not the right option. Modifying DNA. So biomarkers are means not linked to DNA modification. And then sequencing RNA. So biomarkers are not generally used to sequence a DNA. They are used to distinguish or identify, not for the sequencing. They can be used for the comparisons, but not for sequencing. So the right option is option A. Okay, so let's look at the next question, which is what does proteogenomics aim to identify in the context of gene variants? Option A, RNA transcripts. Option B, proteins translated from variant genes. Option C, DNA mutations. And option D, chromosomal abnormalities. So what do you think if we talk about the gene variants? So how the proteogenomics is helping us to identify? Okay, so you both have mentioned option B, so that's right. It helps us to identify the proteins which are translated from variant genes. Okay, so next question is, what is the significance of identifying post-translational modifications in clinical proteomics? Option A, they provide insights into protein structure. Option B, they can affect the protein function and disease states. Option C, they simplify data analysis. Or option D, they indicate gene expression levels. So what is the significance of these post-translational modifications? Yeah. So option B, that means they can affect the protein function and disease states. So this is the right option. And they provide insights into protein structure. So not exactly, because they they will have some impact. They might change a little bit of a protein structure, but they do not provide any insights about the protein structure. And they simplify data analysis, so not at all. They do not simplify analysis because you are also incorporating post-translational modification information. So you are kind of complicating analysis, not simplifying it. And they indicate gene expression levels. So that is also not true because post-translational modification has nothing to do with the expression. So this is also not right. Okay, now let's see question number 17. In clinical studies, what is the role of control group? So to receive the treatment being tested, to provide a baseline for comparison, to analyze genetic data only, or to ensure the random sampling. So what do you think, for which purpose control group is used? Okay, yeah. So the right answer is option B, which means to provide a baseline for comparison, because we already know that in any of the experiments we take a control and a test. So control is just to treat it as a reference or as a baseline so that we can compare it with our test samples. So in similar way, in clinical studies also we have a control group and the another disease group or other group, which is generally means for the comparison sake we take it to receive the treatment being tested. So no, control group will not receive any kind of treatment. To analyze genetic data only, this is also not true, and to ensure random sampling, not at all. Yeah, so I will also want to tell you that in this case they mentioned clinical studies. So in the previous question, if in place of clinical trials, if clinical studies would have been written, so that would also be kind of correct option other than the cohort study. So next question is, why is gene annotation a challenge in identifying novel gene expressions? Option A, it is not relevant to expression studies. Option B, many genes remain unannotated or poorly annotated. Option C, it complicates the data collection process. And option D, it is easily achievable with the existing databases. Okay. Yeah, so you are right that many genes remain unannotated or poorly annotated. So it's true because it's a bit difficult to annotate any gene at mostly at the level of functions. So to predict that whether it will, what will be its influence on the expression, it's difficult to annotate it. So many genes, they remain either unannotated or poorly annotated, and then it is not relevant to expression studies. So this is not true because if you have the gene annotation, then you can means go for the expression. You can compare the expression among different genes. You can study their expression. So this is also not true, and it complicates the data collection process. although it will complicate, but you do not have the information, so it's difficult. So that's why this point does not come into play, and it is easily achievable. So this is also not true, because if you have a novel gene, then you do not have already available database which contain that information. So one more point which I want to add here is that because of this limitation, what happen is that if you have a gene, uh, means currently experimental studies are done, mostly means knockout studies, or all those studies are done to identify the function of particular gene. So in some scenarios what can happen is that in one gene can give rise to multiple proteins, which we call as isoforms of a same gene. So those isoforms can also have different functions. For example, if one protein is involved in some role, for example, there is a gene called PKM, protein kinase. So it has two different isoforms. One is expressed in the embryo and another is expressed in the adults. So if in adults the isoform gets switched, if in adults the embryo one isoform which was expressed during the early stages, if that isoform expresses, so it leads to cancer. So in that case, what can happen is that that gene, its function is only one single function is annotated to this gene, but its isoform, they have different associations, they have different functions. So in that case, till now also we do not have that much proper annotations available. We only have annotations at the level of genes and not at the level of proteins. So in some cases, what you will find is that if you search for two different proteins, for example, in UniProt, then the functional annotation will appear same for both the proteins. So it's very hard to find out the proper functionality of a particular gene. It's very difficult to annotate it actually, because its proteins can have variable expressions or variable functionalities. So that is also one of the challenge. Then next question is, what is the primary goal of variant calling in sequenced transcript data? Option A, to quantify gene expression levels. Option B, to identify genetic mutations and polymorphisms. Option C, to analyze protein structure. And option D, to measure RNA stability. So in the classes you have seen the variant calling format files. I think Kelly has explained. So now you have to tell that what is the primary role of variant calling and sequence transcript data. Okay, so you both of you are right. So it is generally used to identify the genetic mutations and polymorphisms, because it do not tell about gene expression levels. So because by looking at simply at the variant, you cannot quantify gene expression levels. For that, you require experimental evidences. So by just looking at variant, this option is not true. This is also not the primary goal at all. To identify genetic mutation and polymorphism, yeah, it's the right option. Then to analyze the protein structure. So by just looking at the variant calling, you cannot comment on protein structure. Although the variants can influence the protein structure, but you cannot analyze it by just looking at variants. And then to measure RNA stability. So this is also not true. By looking at the variants, you cannot measure the stability of RNA. For these analysis, you require the additional experiments or additional data sets to means identify that whether those variants are linked to stability or not. Okay, next question is, which software is commonly used for variant calling from RNA-seq data? BLAST, STAR, GATK, or CLUSTAL-W? So I think you might not be familiar. Okay, you both have mentioned. Option C. Yeah, so this software is right. GATK is a software which is commonly used for the variant calling from RNA-seq data, and not the BLAST, STAR, or CLUSTAL-W. So BLAST, as we all know, that it is for the pairwise sequence alignment, or if you want to identify given sequence in the already available BLAST database, so that for that purposes you go for a BLAST. It is simply the alignment-based search tool. Then then comes STAR. So this is whenever you go for RNA sequencing and you're analyzing the RNA-seq data. So in that scenarios, you use this particular, you can call it as a tool or a method which is present in the R programming. So this is generally done for the alignments, read alignments to the entire genome assemblies, and then identifying to which particular transcript that read belongs to. So for that purpose, it comes under RNA-seq analysis. And then cluster W, so it is for the multiple protein sequence alignments, or yeah, for those purposes, whenever you want to align multiple protein sequences, you want to compare the evolution history, you want to check that how much similarity or identity is present among different protein sequences, then you use cluster W. Nowadays, I think it is replaced with cluster Omega tool. It was the older version, which was cluster W. Yeah, so option C was the right. Now, question number 21. What can lead to false positives in variant calling from transcript data? Option A, high quality RNA samples. Option B, low sequencing depth. Option C, robust bioinformatics tools. And option D is accurate reference annotations. So, what could lead to false positives? Okay, Ptolemy, do you want to make any guess for this? So, the correct option is option B, which is the low sequencing depth. Yeah, so when we say false positive, what does false positives means? That in some scenarios, what can happen is that um, something which is not exactly true, but it is appearing to be true. That is referred to as false positive, that you are labeling it as positive, but actually it is not positive. So false positives can arise if you have a low sequencing depth. Uh, Means when we say depth, that means you have very lower number of sequences which are present. For example, in RNA-seq, we give emphasis on the number of reads which we obtain. So if I only have a one read, so with that single read, I'm not that much confident enough that this particular transcript is available. So if more number of reads are available, it will increase the confidence, and it will also let me know that this particular match or this particular peptide is not generated randomly, or not generated as a noise. So this is means confidently generated peptide sequence or confidently generated read, RNA-seq read. So in that scenarios, if the depth is high, so it will be a true positive. But if depth is very low, then it might lead to false positives. So there are chances, we can say. So high quality RNA samples, no. If we have high quality RNA samples, then they won't lead to false positives. Robust bioinformatic tools. So if tools are robust, then they will help us to find more true positives instead of false positives. Accurate reference annotations. So if we have accurate reference annotations, then we won't go into the false positive detection. Or we can say that accurate reference annotation has nothing to do with the variant calling from transcript data. These are just for the comparisons. For assembly, we use the reference. So in that way, even if we have accurate reference, but we do not have RNA-seq data which is properly taken, then it will means it has nothing to play with the false positives. So low sequencing depth is the right option. Now next question is, how do splice variants affect the interpretation of transcript data? They have no impact on variant identification. They complicate the detection of mutations in coding regions. Option C, they simplify the variant calling process, and they are easier to identify than single nucleotide changes. Okay, okay, let's see. So yeah, you both are right. Option B is the correct. They complicate the detection of mutations in coding regions. So splice variants are the same thing, which means that a single gene, when it converts into the mRNA, so that mRNA is premature mRNA, and it ultimately its modification is done, its processing is done, we can say. So in that, capping, tailing, and then splicing is also one of the steps. So in that splicing, introns are removed and exons are combined. So in some scenarios, what can happen is that splicing is not the exact splicing. There are chances of alternate splicing in which different exons can combine in different combinations, so which can give rise to different variants of this spliced form. So that is known as spliced variants. We call them as multiple transcripts or as isoforms when it is converting to protein. So we call them as isoforms of the same gene. So these splice variants, how they influence, they complicate the detection of mutations in the coding regions, because we do not know that whether it is a mutation or whether it is a splice site change. So that's why this is one of the challenge while interpreting the splice site variants. First option was they have no impact on variant identification, so this was not true because they complicate our analysis. They simplify variant calling process, not at all. They do not simplify at all. They are easier to identify than single nucleotide changes, so not at all. They are not that much easy to identify. They are difficult to identify. That's why identification of splice variant itself is a challenge. If we already have the information of all the splice variants, then it might not interfere with the variant or interpretations. But till now, they complicate the interpretation because we do not have the enough data set for the all the splice variant possible. Next question was, which method can help improve the accuracy of variant detection in RNA-seq data? Option A, using a low quality reference genome. Option B, increasing the number of replicates. Option C, reducing the sequencing depth. And option D, ignoring the batch effects. So, which method you think is helpful to improve the accuracy? Okay, option B. Okay, yeah, so you both of you are right. Uh, Increasing the number of replicates help us to improve the accuracy of variant detection because if more replicates produce similar results, so we will be more confident using low quality reference genome. So as means from this name only we can say that this is not right. Reducing sequencing depth, not at all, because if sequencing depth is low, then it will lead to false positives, which are also not good for the accuracy. Ignoring batch effects, not at all, because we have seen during our previous weeks that batch effects can have the drastic effects on the identification. So we have to remove those batch effects. So that's why we cannot ignore these batch effects. They can have severe outcomes. So the right option was increasing the number of replicates because number of replicates can help us to improve the accuracy. Next question was, why is it important to use a well-annotated reference genome when identifying the variants? Option A, it eliminates the need for the quality control. Option B, it provides a basis for accurately aligning reads and detecting variants. Option C, it is not relevant to variant identification. And option D is it simplifies the data analysis. Okay. Yeah, this time also you both are right. So option B, because it provides a basis for accurately aligning the reads and detecting variants, because we know that for the reference means we require reference because from our already means the data which we already have, so we map it to the reference and then do all the annotations or assembly thing. So if our reference is well annotated, then it will do better alignments and we will be able to detect the variants in means accurately in a better way. It does not eliminate the need for the quality control. So this is also not the right option. And it is relevant for the variant identification if our reference is properly annotated and it simplified data analysis. So it can simplify the process of annotating or process of aligning, but data analysis is another thing. Means we have to analyze that data. So to some extent, this is a bit confusing, but main result will be the option B. Now question number 25, what is a potential effect of large deletion in a genomic region? Option A, improved protein function. Option B, loss of important regulatory element. Option C, increased gene dosage. Or option D, enhanced cellular metabolism. So what is a potential effect? It's not saying that always this effect has to be there, but what do you think potentially can happen? Okay, yeah. So option B is right. That means loss of important regulatory elements. So if there are larger deletions, then they can influence the regulatory elements. And when we say regulatory elements, so these elements are generally associated with the expression of genomic regions. These regulatory elements either could be promoters or enhancers or silencer regions, where the transcription factors or other factors bind, and then they influence the means expression of that particular genomic region. So if there will be larger deletions, so they can delete those regulatory regions. Now let's look at the other options. So improve protein function. So deletions, most of the cases, deletions can never improve the functionality of protein. It can either delete that protein or means, for example, if that deletion is in the you can say silencer region, but that will also be very small region. Larger deletions will ultimately wash away the ability of protein to properly, ability of gene to properly express. But in case if that is in those regions where the means inhibitors bind, then it might improve the function, but we cannot say this option to be right. Then increase gene dosage, not at all, because deletions cannot increase the gene dosage. And then enhance cellular metabolism. So this is also not true. Means it depends. If in very rare scenario, if some of the enhancers which are, so yeah, the another term is okay. So means, for example, if there is deletion in those regions where the we can say where the inhibitors bind or other things bind which do not allow our protein to express, in that scenarios it can enhance, but they are very rarest of the rare scenarios. So ultimately, option B is right. Loss of important regulatory elements, because in regulatory elements only both the all these elements will come into play, which means both the enhancers, silencers, promoters, all those elements come under this term. So this will be the right option. Now, question number 26. What is the potential impact of gene fusion events? A. Decreased transcription. Option B. Creation of novel proteins with new functions. Option C. Increased genomic redundancy. And option D. Normal gene expression patterns. So for this, we have also seen in the previous session of the when we were looking at that chromosomal example, Philadelphia chromosome. So in that we have seen that how two genes get fused together and they ultimately cause a disease. Okay. So yeah, you mentioned option B. So yeah, B option is right, which means creation of novel proteins with new functions. And those new functions might not be useful for our cell, but still it leads to change in the functionality and it creates the new protein. Decreased transcription. So this is not the potential impact. There are chances that it might decrease, but yeah, it will not be considered. Increased genomic redundancy. So we cannot say that because in this case, genes are fusing, two different genes are fusing, but still it will not means if there is duplication, then they can increase the redundancy because redundancy means repetitions. So this gene fusion won't lead to redundancy because that was in that case example we have seen that it was imbalanced translocation. So from one place it is getting deleted. So in that scenario, it is not leading to genomic redundancy. The normal gene expression patterns. So if some gene is changing its position, it will not means still maintain the normal functionality or normal expression pattern. So its expression pattern will change because we have already seen that there are different studies which have mentioned that synteny is very important for the functionality of a gene. Synteny means that in the evolution when we study, we compare the chromosomes among different organisms or different individuals. So how that chromosome means how the genes are mapped to chromosomal positions, what is simply done is that we look for the neighboring genes and then we make sure that okay, in this particular chromosome these genes are neighbor to one another and what is their distance in that particular gene, so that we can means identify which genes are linked genes and which genes are independent, which do not have any influence on expression of one another. So in that scenarios, the genes means their positional distribution is also very much important. So means position of genes can affect the functionality or expression patterns. It can change by change in the position of those genes because their neighboring genes can also interfere with, because there are some cis regulations or trans regulations which are also undergoing on the chromosome. So in that scenario, the expression patterns can change. Now, question number 27. How can mutations in regulatory regions affect the gene expression? So, option A, they always increase expression levels. Option B, they can enhance or repress the expression depending on the mutation. Option C, they have no effect on gene expression. And option D, they only affect the non coding genes. Okay, yeah. So, right answer is option B. Because we have already discussed that regulatory regions means they are regulating the functionality. So regulation can either be positive or negative. So it can either enhance or repress the expression because it will not always increase expression or always decrease expression. So uh, C option was they have no effect because these are regulatory regions, so they will have effect. They only affect non-coding genes. So this is also not true because regulatory regions are generally lie means upstream or downstream of the coding region. So they lie themselves in the non-coding region. They comes under non-coding genes. So they affect the coding genes most of the cases. Okay, question number 28. Which of the following genomic changes is most likely to cause a frame shift mutation? Option A, insertion of a single nucleotide. Option B, deletion of three nucleotides. Option C, substitution of one nucleotide. And option D, duplication of a gene. Okay, so Polemy has mentioned option B, but the right option is option A, which is insertion of a single nucleotide. Okay, I have not mentioned it. Let me explain. So, what does frame shift mutation means? So in the previous session, as we were looking at the integrative genome viewers, in that we have seen that entire chromosomal nucleotide sequences were given, and in that we have seen that if we go for three-frame translation, then in 3 frames that particular DNA sequence can be translated. So what we mean by 3 frame, that means if we start from the +1 position, second if we start from +2, and then if we start from +3, or we can say from 0, +1, +2, or any way you can say that. So what can happen is that if you delete three nucleotides, then ultimately the frame will remain the same, because those three nucleotides, means it depends actually, because those three nucleotides, if I will read it from 0, 1, 2, or 1, 2, 3, so in that case, if three nucleotides are removed, that means that amino acid is removed, so frame will still remain intact. So in that case, it might not lead to frame shift mutation. So in this case, deletion of three nucleotides, in some scenarios, it can lead to frame shift, but means there are possibility that it might not lead to frame shift mutation. That's why this option was not true. Whereas if there is insertion of single nucleotide, then it will 100% always lead to frame shift mutations, because it will change the frame. For example, if you read 1, 2, 3, and in third position, if new insertion is done, so now from 4, 5, 6, the new nucleotide which has come up in that position, so now that will change the entire frame. So amino acid sequence might get changed. So three nucleotide deletion in some scenarios, because those three nucleotides are giving rise to one single amino acid. So even if those three nucleotides are deleted, still it might not lead to change in the sequence of amino acids or in the frame it was being read. Substitution of one nucleotide. So even if you substitute one nucleotide, it will not change the frame. It might change the amino acid sequence, but it will not change the reading frame. Duplication of gene. So this has nothing to do with the frame shift. Even if that gene is duplicated, but still its reading frame is conserved. So this is also not true. Insertion of a single nucleotide is the right option. Deviation of amino acid. So deviation means change in amino acid. Are you asking about the substitution of one nucleotide? In which perspective you are asking for the deviation of amino acid? In the order? So, which I have mentioned here, or you are asking some other question? Because in this perspective, the deviation of amino acid means that if a single nucleotide is changing, then that single nucleotide change can also lead to missense mutation, in which it can means produce a different amino acid sequence. Because frame is different thing and amino acid sequence is different thing. So when we say frame, we means in the reading frame, reading count. Okay, due to genomic change. Yeah, so due to change in the genome or genomic sequence, it can lead to change of amino acid as well. So deviation of amino acid means change in amino acid. Mostly deviation is not the right word, but still if we say deviation of amino acid, it means change in amino acid. And yeah, genomic changes can lead to this deviation of amino acid. But in other scenarios, it might not deviate, because in synonymous mutations, even if genomic change is there, but still it do not change the amino acid. So both the things exist. Yeah, I will just check if I can write. I've never tried this. Okay, so for example, I have this gene, very difficult to write. Okay, so for example, I have this gene sequence, and this is the start site from where I'm starting to read this gene. So this could be first position, this is my second position, and this is my third position. With position, I mean nucleotide position. This is my fourth nucleotide. This is my, sorry, fifth nucleotide, and this is my sixth nucleotide. So when I read in there are three frames which are possible. Only in three frames we can generate different sequence for this particular gene, means the protein sequence, in three different ways. If I start from here, then this will be one of my protein, one of my amino acid. This will be my second amino acid, and similarly for seventh, eighth, and ninth position. Sorry. Yeah, so similarly for my seventh, eighth, ninth position also, same thing will happen. That will be my third amino acid. Now, in case of second reading frame, let me change the color. So now it will start from here. Now these 2, 3, 4 nucleotide will be making my first amino acid. 5, 6, 7 will make my second amino acid. Similarly, in case of um, third reading frame, it will start from third position. So 3, 4, 5 will make my first amino acid, and then 6, 7, 8 will make my second amino acid. Now, if I start with my fourth reading frame, so which means it will take into account my fourth position, and it will synthesize amino acid from 4, 5, 6, which will be exactly same to that of my first reading frame, because in first reading frame also I have 4, 5, 6. So in that way, frame of reading, that means in the triplet, means position wise triplet I'm reading, so it will same as my first frame reading. So in that way, uh, it will generate the same frame. So that's why I do not call fourth position or fourth reading frame to be a different, means giving rise to different sequence. It will generate the same sequence. It will only miss the first amino acid information, but the coding frame will be intact. So that's why even if I delete three nucleotides, for example, if I delete these three nucleotides, then 4, 5, 6, still the coding frame will remain maintained. So that's why deletion of three nucleotides was not most likely to cause a frame shift mutation. But in some scenarios, it is possible. But if I go for all these three frames, then it will not get interfered even if I delete three nucleotides. Okay, yeah, it was horrible. Sorry for this. Okay, next question. How can genomic change and how can genomic changes lead to evolutionary adaptations? Option A, by creating mutations that are always harmful. Option B, by providing genetic diversity that can be beneficial. Option C, by stabilizing existing traits without change. And option D, by preventing new mutations from occurring. So how they can lead to evolutionary adaptations? Okay, option B. Yeah, this is somehow related to our evolutionary study classes. Okay, you're right. Option B is the right option, because if some genomic changes are providing some beneficial advantage, then they are most likely to get adapted. Okay, so first option was by creating mutations that are always harmful. So if some mutation is always harmful, which means harmful means it is either fatal, or it is causing death of individual, or it is leading to poor survival. So in that scenarios, you can think that if some mutation is harmful for us, if people are dying due to that, so why that mutation will be favored? Because it will ultimately lead to vanish of that particular population. So in that case, it will not be a evolutionary adaptation. It will not be adapted. It will get lost by stabilizing existing traits without change. So this is also not true, because it is not stabilizing existing traits. Genomic changes have some influence. So they do not stabilize it. They kind of provide us some advantage so that we can adapt to new changes. That's why these are mostly favored. by preventing new mutations from occurring. So no genomic mutations, because these mutations are very necessary. Because if mutations will happen, then only we'll evolve to better individuals, we'll evolve better. So that's why mutations are very necessary. Means they are also necessary. Not all mutations are harmful. So in that scenarios, mutations are also helping us to adapt to new changes, new environments. So yeah, mutations are useful also. So if those are beneficial, then they will be adapted. Next question, what distinguishes germline mutations from somatic mutations? So option A, germline mutations affect somatic cells only. Option B, germline mutations can be inherited while somatic mutations cannot. Option C, somatic mutations are always harmful. And option D, germline mutations occur after fertilization while somatic mutations occur before. Okay, so you mentioned option B. Yeah, so option B is right because we know that germline mutations, these are happening in the germ cells and they can be inherited, while somatic mutations, they cannot be inherited, which are occurring on the other cells other than the germ cells. So they are generally not inherited or not passed on to other generations. And we cannot say that germline mutations affect somatic cells because they do not affect somatic cells, they affect germ cells. Somatic mutations are always harmful. So this is also not true because some mutations, they might not be harmful. They can be a passenger mutations or other kind of mutations. So they are not always harmful. They can be harmful in certain scenarios, but not always. Then germline mutations occur after fertilizations. So this is also not true because uh, while somatic mutations occur before, so fertilization has no role to play here in case of somatic mutations because these are occurring on other cells of the body which are not involved in fertilization at all, and they have nothing to do with the either they occur before or after. They can occur at any time. Okay, so next question was, which of the following is an example of a consequence of germline mutation? Option A, a skin cancer tumor. Option B, an inherited genetic disorder. Option C, a localized infection. And option D, a response to environmental stress. So yeah, option B is right because we have already seen that germline mutations are means involved with passing of a disease or inheritance. So they're linked with inheritance. So and mostly the skin cancer, these tumors, they are mostly so linked with somatic mutations. Localized infection is most probably linked with the somatic mutations, and response to environmental stress is also linked with the somatic mutations, not an consequence of germline mutation. Now question number 32. What type of mutation would be most likely to contribute to the development of cancer in an individual? Option A, germline mutation in all cells. Okay, what is localized infection? Yeah, so when we say localized infection, which means the infection which is localized to particular tissue or particular organ, so that is referred to as localized infection. Yeah. For example, skin cancer tumor. So these tumors are also restricted to one particular location, but when it enters the blood or when it is metastasizing, when there is metastasis of that particular tumor, sorry, that particular cancer, so in that case, it is not localized to one particular part. So that is known as, means that will not be localized, but localized infection means it is just local to one particular part. And in this case, they have mentioned infection. So infection can be any kind of infection, not exactly cancer. Yeah, but it is restricted to one particular organ. So yeah, so that is not the consequence of germline mutation. Yeah, so the next question was, what type of mutations would be most likely to contribute to the development of cancer in an individual? Option A was germline mutation in all cells. Option B was somatic mutation in a specific tissue. Option C, silent mutation in a gene. And option D was mutation in a known coding region. Okay. So the right answer was option B, which is somatic mutation in a specific tissue. So which is most likely to contribute to development of cancer. Because if germline mutation in all the cells, so this is not true because germline mutation cannot happen in all the cells, and if it happens in all the cells, then person will die because so this option is not true at all. Somatic mutation in a specific tissue, so in specific tissue it will happen, then it can lead to the development of cancer. For example, in the previous question we have seen about skin cancer tumor. Next option was silent mutation in a gene. So if mutation is silent, so silent means it is not leading to change in the amino acid sequence. Means although nucleotide change, but amino acid sequence does not change. So in such scenarios, first thing which you can get from this point is that silent mutation will always come when it is in the coding region. Then only you will be able to identify whether it is a silent mutation or non means missense mutation. So silent mutation, which is not changing the amino acid. In such scenarios, it's very unlikely that it will develop to a cancer. It might not influence the uh, protein sequence. In some extent, it might change its means expression to some extent, but that is also not true. Most of the cases that do not influence the protein at all. Then mutation in a known coding region. So if mutations are there in the known coding regions, then they can influence the they can influence the regulation of the protein, but they are not most likely to contribute to the cancer. They are less likely than somatic mutation. So in this case, somatic mutation was more likely as compared to mutation in a known coding region. But we cannot ignore this option because in some scenarios it can have influence on the particular protein expression and which might be linked to the development of a cancer, because if that particular protein is either overexpressed or not expressed, then it can be linked to a cancer. But in these options, B option was most likely. That's why it was correct. Now question number 33. In what way can somatic mutations contribute to cancer progression? So option A was by increasing overall fitness of the organism. Option B was by causing changes in the genes that regulate cell growth and division. Option C by being inherited by future generations, and option D was by affecting all tissues uniformly. Yeah, so you are right. Option B is the right answer because these somatic mutations, first of all, they are mentioned by increasing overall fitness of the organism. So somatic mutations do not increase the overall fitness. Means in very rarest of the rare scenarios, it might increase fitness, but no, this was not true by increasing overall fitness. So, and option C was by being inherited by future generations. So this is also not true because germline mutations are the one which are inherited and not the somatic ones by affecting all tissues uniformly. So this is also not true because somatic mutations in most scenarios they influence one particular tissue or localized kind of infection is mostly observed because whenever a person is means exposed to one particular environmental conditions or some other exposing agents or some other agents which which give rise to these somatic mutations. So in that scenarios, it does not affect all the tissues uniformly. Whenever we'll be talking about these mutations, so they are not uniformly happening in all the cells. They are very specific to some particular cells from means from some tissues. So yeah, it will not influence all the tissues uniformly. So the right option was by causing changes in the genes which regulate the cell growth and division. So most of the scenarios they influence these, they regulate these genes, and in other scenarios they might influence other gene as well. But most of the genes are cell growth and division because if those genes are not properly regulated, if cell growth is not balanced, then it will lead to means unlimited growth of cells, which will lead to tumor. Similarly for the division as well, and other genes which also come into play are the apoptosis genes. Because if your particular protein is means dividing infinitely, it is dividing some cells. So then if there is no proper apoptosis, so there will be no cell death, which will also lead to uh, tumors or cancers. So other gene categories will be the apoptotic genes, and then the third category is the tumor suppressor genes. So if those genes are repressed or silenced, if their regulation is also changed, then they will also lead to cancer. Similarly, if apoptosis genes are their regulation is influenced, then also they will lead to cancer, and these cell growth and division genes are also there. So any option can come in your exam. So in this scenario, this option was right. Now let's see next question. So what is a common source of somatic mutations? Because these somatic mutations, there must be some source. They're not automatically happening because there are some neutral modifications, you can say, which are undergoing, which are already going due to replication errors or other things. So there has to be some sources which are giving rise to these somatic mutations. So option A is environmental factors such as UV radiations or chemicals. Option B was inherited genetic predisposition. Option C was errors during DNA replication in the germline, and option D was transmission from one organism to another. Okay, so Polemy has mentioned option A. Yeah. So option A is right. Environmental factors such as UV radiations or other chemicals, which are the common sources of somatic mutations. Then inherited genetic predisposition. So this is also not true because somatic mutations are not inherited at all. Errors during DNA replication in the germline. So this is also not true. Although DNA replication can also produce somatic mutations, but in the germline they have mentioned, that's why this option is not right. Then transmission from one organism to another. So as we know that these mutations are not transmitted from any individual to any other individual. So first option was the right one. Now question number 35. So how can somatic mutations influence the treatment of cancer? Option A, they provide a predictable response to treatment. Option B, they can lead to resistance against specific therapies. Option C, they are irrelevant to cancer treatment. And option D, they simplify the diagnosis of cancer. So, how these somatic mutations can influence cancer treatment? Okay, yeah, so option B is right. They can lead to resistance against specific therapies. Option A was they provide a predictable response to treatment. So, first thing is that we cannot predict the response by means simply looking at the mutation that this will if we treat it with this particular reagent or this particular drug then this will be the predictable response. So we cannot predict response so much accurately. So this is also not true. They are irrelevant to cancer treatment. This is also not true because somatic mutations are important. They can give us many insights, so they are not irrelevant. They simplify the diagnosis of cancer, so they do not simplify at all, and then they can lead to resistance against specific therapies. So yeah, this is true because they can generate some resistance in which therapies might not work. And one another thing which I want to add is that there are multiple mutations, so to identify which mutation is linked to a cancer it's very difficult. So it's very difficult, that's why they lead to resistance against specific therapies, and specific therapies they might not means act in that way in which we expect it to work. So in that way, they influence the treatment of cancer. This was all about today's session. So if you have any questions regarding the assignment or regarding this week, so you can ask if you have, because we still have a few minutes with us. Okay, the identification of database for new variant is difficult. Uh, New variant means transcript variant, or what kind of variant you want to ask? Because new variant databases identification is not that difficult. We can devise because proteogenomics is helping us in that only. With the customized generation of the protein sequences, we can easily identify the new variants. Yeah, I will provide you with the link of those papers in the end of the presentation. I think in the last session you have asked about those red blocks with the star. So I have also added that slide into my previous presentation. I hope you have gone through that slide. Yeah. So what I came to know is that that was the previous representation, or might be the offline mode of that app, in which they do not use to write the stop codon, they used to simply mark it with the star. Yeah. So I will mention these papers. Catalogs to identify novel peptides, or we get the functions. So I do not get your question exactly, but Uh, Miss catalogs, with catalogs, what do you want to say? Because novel peptides we first identify, then we do the cataloging. So cataloging is kind of you're managing some record. That is known as cataloging, I guess. With cataloging, what I mean with cataloging is that you have the information and you are kind of storing it or you are managing it. That is known as cataloging. You are preparing catalogs. So if you have the information of novel peptides and you know from which particular transcript it might come from, so you catalog that information, you store that information. So according to my perspective, I feel like that is cataloging. Yeah. Yeah. So I think we first identify those peptides and then we catalog it. Because cataloging is a journal term. Because if you are using the already designed customized protein sequences, then that is also one of the catalog from where you are mapping the means peptide spectra and you are matching, you are finding out what could be the possible match to that peptide spectrum. So novel peptides are also kind of identified from there as well. So it depends that what is the ultimate aim. So if we want to means attach the functions to the novel peptides, then yeah, firstly we need to identify those novel peptides and then take the experimental data sets which are already available, which have already marked the functional aspect to one particular peptide. Then we can do this association of the novel peptides with their functions and then ultimately catalog them. Yeah, novel peptide identification is difficult if database is not present. Why not in case of genome or RNA level? First of all, at genome level, in genome level, if you have a genomic sequence, because genome sequence is constant thing, it does not change. But if we talk about RNA level, then it is also difficult because in RNA also there are possibilities that different splice variants can arise. So in that case, even for the RNA also RNA sequencing, it is a challenge. So, because RNA spec might not, when you are going for assembly of RNA sequencing reads to the genome, so uh, there is also a difficulty to identify that what would be the sequence of this particular RNA. To assemble it also means assembly of the RNA sec is also a bit a challenge to identify that whether, because in many cases we do not have proper annotations, because as we have seen with the proteogenomics, you can modify or you can refine the gene models. So gene models means when you have the information of all the coding, non-coding elements present in the gene. So um, because in the genome there are some cases where we do not have proper labeling of all those coordinates, all those exons properly. So in that case, even if you are going for the RNA sequence analysis, then also you will face this challenge. So with RNA level, you will be able to identify new transcripts or new splice variants. And then ultimately, with that particular information in your hand, you can synthesize uh, possible means proteins which can be synthesized from it. And ultimately, novel peptide identification will be helped with the help of those predicted RNA sequences. So one means main answer is that in case of genome, it is constant. That's why we do not face challenge, but in RNA, we still face challenge. How to deal with novel altered splice junctions? So novel altered splice junctions. So first of all, as we have discussed about in the RNA-seq only, that there is a possibility multiple splice variants could be present. So in our current databases, which we already have, we do not have information of all the splice junctions because for the splicing, the main protein which is important is spliceosome. So in many scenarios, what can happen is if there is, I mean, spliceosome is not that efficient in splicing, then it can alter the splice junctions. It can either lead to the means inclusion of splice means N-terminal region of the intron into the exon while splicing. So that can that in that case it will vary the splice junction. For example, normally if splicing happens, then it will chop off my intron exactly from the 5' and 3' splice site. But in some scenarios, due to the problem with the spliceosomal machinery, it can lead to the change in the splice sites where it cleaves. So in such scenarios, if splice site changes, so junction will also change. So splice junction means when the two exons are getting linked. So in that case, the junction of that means a splice site. So due to change in the splice sites which are cleaved, the junctions will also get changed. So in order to deal with those novel junctions, first of all, we need to first identify that which are the proper exonic and intronic boundaries. If we know about the boundaries, then we will be able to identify the novel altered splice junctions. Because then we can design the customized databases that what are the possible protein sequences which can be generated. So that thing can be done. That can be done to means deal with these novel altered splice junctions. So for this, firstly, we need the information from the either from the RNA-seq or from the peptides. Then only we can do this. We cannot go theoretically to identify the novel altered splice junctions. But if we have the RNA-seq data, and if we during mapping, if we see any changes, then we can map it. We can see that okay, this is my these are my introns, and this is my exon, this is my intron site. So if I'm getting RNA-seq which is mapping to, which is covering some of my intronic regions, then I can say this is the altered splice junction. So with RNA-seq, I can identify the altered splice junctions. And with RNA-seq, means after getting altered splice junction, I can design the means customized protein sequence without going to experimental level, and then I can identify the peptides which might be specific to that particular altered splice junction region. So yeah, RNA-seq is important for identifying this. Yeah. Okay, by the label-based and unlabeled technique, can we quantify the phosphorylated sites of proteins? Okay, so phosphorylation is a kind of a post-translational modification. So first of all, before entering into label-based and unlabeled techniques, we have already seen that even with simple mass spectrometry also, uh, even if there is phosphorylation, then mass to charge ratio will change. So I think even if you don't do label-based or unlabeled, then also you can quantify the phosphorylated sites because they will ultimately change the mass to charge ratio. Because with the phosphorylation, they will add on some mass to it. So mass to charge ratio will change. But if you have exact information, then means you can quantify it, but to identify, yeah, identify also, you can also identify those sites as well. Because after the fragmentation results, you will be able to know this, even exactly know the positions where that phosphorylation is happening. Yeah, so label based and unlabeled technique has nothing to do with the phosphorylation. Means you can do it without those techniques as well. This only I want to mention. Is there any question related to this in the assignment? Three and 6 feet. I think I'm not able to get it. What does this mean? Need of 3 and 6. Okay, frame translation. Okay, sorry. Yeah. So, so fun. Actually, there is no such need. First of all, what we do is by doing 3 frame or 6 frame translation, we are just trying to find out that what are the possibilities in which one gene can give rise to multiple protein sequences. So this is just to find out that how many different version of a protein sequence can be possibly means present. So generally, why it was means identified because in the previous time when we were having the genome sequence, so it was very difficult to identify the protein sequence by experimental things. So in that case, what people used to do, they used to take a genome sequence and they used to predict that what protein sequence can be produced from that. So in that scenario, 3 frame translation into came into sorry 6 frame translation came into play because we know that the DNA can be translated on both the directions. Means it can be read from forward direction also and reverse direction also. So, and another information was that the genetic code was also devised. So in that case, we came to know that three nucleotides generate a single codon and which give rise to amino acid. So which means that in a frame, if I am starting from my first position, then first three nucleotides will give rise to one amino acid. So if I read the frame like 1, 2, 3, then 4, 5, 6, then 6, 7, 8, 9. So that will be a one frame in which one sequence of protein can be constituted. Next possibility is that if I do not read from my first position, I start from second position. Then I will check for the protein sequence. So it will generate a new protein sequence because now my frame is shifted by one nucleotide position. So now it will start with 2, 3, 4, 5, 6, 7, then 8, 9, 10. Now, the third possibility is that if I start from third position, then also a new protein sequence can be synthesized if I'm reading it from the forward direction. But what happened is when we start with the fourth nucleotide position, then it will again generate the exact same sequence as generated by the first frame. The only difference will be that it will miss the first amino acid. So in that case, it will not generate any change in the sequence. That's why three frame translation came into play, that in the forward direction, three different proteins can be, synthesized. Three different proteins can be synthesized. Similarly, from reverse direction also, we can synthesize the protein sequences. That's why from reverse side, if we start from the first position, that will be my fourth frame. Similarly, from second position, if I read, it will be my fifth frame, and similarly from third position, if I read from the reverse side, so it will be my sixth frame. So in all these six frames, these are the six possible variants of protein sequences which can be generated. But this is not always the case, because now we have seen that the machinery is quite complicated, because alternate splice variants can give rise to multiple diversity of protein sequences. So this was just for the theoretical purposes, that what are the possibilities. But this is not means we cannot say that this is it. There are mutations, other things also which are going on, but this was the main concept behind the three frame and six frame translation. Okay, thank you so much. So if there are no further questions, then I will end the session. Okay, thank you so much.